is Lab 5, Microscopy, where we will be going over the parts of a microscope and looking at some of the functions and procedures for a microscope. Okay, we're going to hit a few of these topics here, and then some of them we won't be able to do because we're doing it on the video. Okay, so but we do want to go over a little bit of that. So microscopes are a really important tool for scientists because it allows us to see things in more detail, and also allows us to see things that are too tiny, too small for us to see with our naked eye. Okay, so a lot of things that we think about for biology are too small for our naked eye. So we're going to talk about briefly 3 different types of microscopes. And then we're going to go over a little bit more about the compound one because that's the one that we would have been using in lab here. Okay. So the first one is a dissecting microscope, and it's used to magnify a whole specimen. Okay. And it's going to magnify about 10 times. Okay. So make it 10 times bigger, and you're going to look at an entire specimen. When you look at that. So kind of think about like binoculars, okay? And so I'm going to hold up one, okay? Here. Here is a dissecting or stereoscope, okay? Dissecting microscope or stereoscope. And I have the whole specimen here. It's a honeycomb, like a wasp's nest, okay? And then so I would have it here, and then you would look through the binocular, okay, biocular stereoscope, 2 eye pieces, and you would just look through, and it would magnify it 10 times as you go through. Okay, so set that to the side. Okay, so this will be one where you look at the whole specimen. The second one is a compound light microscope, and I'm going to rearrange a little bit here. Okay, because I'm about to set up a table. This is going to be used to see a specimen that is really not visible to your unaided eye. So if I look at that, I'll be like, what is that, right? You can't really see that, okay? And it's going to magnify somewhere between 40x to ours is 400x, okay? And there's a couple of different models of microscope that can have a little bit higher magnification, but the one in our lab goes up to 400. Okay, so it's going to be 400 times bigger, and we're going to get to see that in just a second. I'm going to pull up a slide on the microscope and project it up here before we're done. Okay, and this one, like I said, we're going to be able to see maybe like a cheek cell or something very tiny to magnify it to see a little bit more detail. Okay, we would see the outline, the basics of that cell. The third type of microscope is an electron microscope. Okay, and this is where we're going to see very, very, very tiny specimens. Okay, ones that are 0.2 micrometers. Okay, 0.2 micrometers, very, very small. Okay, 200 nanometers. Did I do that right? Did I do my math right? You guys look back at the metric measurement lab and make sure that conversion is correct, okay? Or you're moving that decimal. So these are very, very tiny specimens, okay? And usually when you have these, there's actually 2 different kinds of electron microscopes. There's a scanning electron microscope, or a SEM, okay? And there's also a transmission electron microscope, or TEM, okay? And they're going to look at different areas of that specimen, okay? The scanning is going to scan the surface, okay? So it's going to send an electron beam to it, and it's going to see the surface. What does it look like? Is the surface smooth? Is it rough? Does it have holes in it? Does it have protein sticking out? Okay, it's going to look at the surface, right? Scanning and surface, S&S go together, okay? The transmission one is going to send that electron beam, okay, through there, through the specimen to look at the inside. So you'll be able to see more detail of the nucleus or the DNA or the mitochondria, okay, what's inside of that specimen, okay? And so this is going to be utilized to see something that's very, very small, okay? We don't have one of those here in our lab, okay? They look a little bit more like a computer, and then you put the specimen in and then you would pull up a computer image for those. Okay? All right. The next section, 5.1, is care of the microscope. Right? Obviously, you probably don't have a microscope at your house. Okay? But you would care for a microscope like you would like your glasses. You would use a special kind of paper to wipe the lens. You would take care not to break them or to drop them. Okay, things like that. Or your iPhone or tablets or something like that. You would take care to make sure that you're taking care of those. Okay. So procedure 5.2. Okay. Underneath all of these labels is a picture of a microscope. Okay, yours is a little bit bigger. I made mine small so I could pull it up a little bit on here so you can see it. Okay. While we're going through this, I highly recommend that you label it. Okay, that you would draw lines and then you would label that as we go. Okay, because you will be tested, quizzed on parts of a microscope. Okay. All right. So let me set this table up real fast so we can get the microscope in the screen. Table's heavier than the microscope. Make sure we're in view. Give us just a second to make sure. All right. Okay. So we're going to have these parts of the microscope. So we tried to put this in the same orientation as it is for your picture. Okay. So 5.2 goes over the parts of the microscope. Okay. The first part is the arm. Okay. It's this right here. If I turned it, it's going to have a red handle. Okay. And this is where you would hold it when you carry it. Okay. You're obviously not carrying it at home, but for your information. Okay. So that's the arm. Okay. The next one is the base. Okay. The bottom is the base, right down here. Okay, that's the base. It's going to be the platform of which it sits on. The next one is the stage. Okay, this is the stage. This is where you're going to put the specimen that you're going to look at, or the slide that you're going to look at. So that's right here, this little platform right here. Okay, just as if a performer is going to go on stage, your specimen's going to go on stage. Okay. The next one is talking about controls for that stage. They're on this side, that's why we turned it that way. Okay, there are two knobs right here that are hanging down from the stage. Okay, one's going to move the specimen side to side. Okay, so there's some clips, some little things on the top, and it's going to move these side to side. And the other one is going to move this front to back. Okay, front to back. So you're just going to be able to manipulate, hey, I want to look over there. Hey, I want to look up here. Hey, I want to look back there. Okay? So you're able to move that. Okay? The next thing you want to do is you want to be able to focus. Okay? So you have your specimen on here. You've moved it where you want it to be, okay, with these adjustments. And now I want to focus. Okay? There are two knobs here, a larger one and a smaller one. Okay? The large one is the coarse adjustment, and when I turn this, okay, I want you to watch the stage. It's gonna go down. Okay, so you're gonna be able to move that stage farther away to focus, probably not this far, or closer to focus. Okay, so when I turn the larger one, the one closer to it, in the back, it moves the stage a lot. Big increments at a time. Coarse, okay? Coarse as in rough, okay? Rough adjustment. The smaller knob, I can spin it, and it barely moves this stage, okay? I'm going full turns, and it's not moving near as much, okay? So this is moving and focusing in very tiny increments as you go to focus, okay? So I may look in there and it'd be focused for my vision, okay? And then I go, hey, look at that. And you look in there and you go, whoa, it's just a little out of focus. You would take the fine and adjust it just a little bit, right? It's like if you go to the doctor and they're doing glasses and you're like, this one or this one? This one or this one? So they're giving just a little bit of tiny adjustment for you there, okay? So that's the coarse adjustment and the fine adjustment. The next parts of this microscope are the lens system, okay? And we're going to start the objective lenses, okay? And those are right here, hanging right here, underneath. There's 3 of them, okay? I usually start up here, that's why I paused for a second, right? So these are the objectives. They're close to the object that you're looking at, okay? So the objective lenses. There are 3 of them, okay? There's a small one, and then a medium-sized one, and then a larger one, okay? A larger one here, okay? So there's 3. A small one, this is the one that you're gonna start looking at your specimen using, okay? It's called the scanning. You're gonna scan that area that you can see, okay? And then you're going to find your specimen on there, okay? We'll talk about field of view and magnification in just a second, okay? So you're gonna start on the small one. This is the scanning objective. Then you would move to get more detail, more magnification on this one. And even though it's in the middle size, okay, it's called the low magnification or the low power objective, okay? And then you would go to the highest power, the largest one, okay? And it's gonna have the highest magnification, okay? So start at the scanning, then move to the low power, and then you would go to the highest power, okay, for looking at your specimen. We'll talk about magnification just again in a second. The other part of the lens system is the ocular lenses. This is where you would look with your eyes, okay? And so if I held this one up, the stereoscope, okay, it's gonna look like this at the top, okay? Where you would put both your eyes, one eye here, one eye there, okay? And so this is the ocular, okay, like the eyepiece, okay, and you would look in. You're able to adjust them and put, just like you would binoculars to fit around your eyes, okay? These are gonna magnify 10 times. This whole microscope magnifies 10 times. This is the stereoscope, the dissecting scope. It's just these. If you look under here, there's no objectives. You just look straight down, it magnifies it 10 times, and you see the image, okay? In the light microscope, the compound light microscope, you look in, it magnifies it 10 times, and then it's going to go through one of these objectives, and it's gonna magnify it some more, okay? We have a chart later that will go through that, okay? So 10 times, and then some more, and then we'll continue to talk. The next thing you wanna be able to do is adjust the light, okay? You wanna be able to adjust the light to see your specimen, and also to your sensitivity of your eyes. So underneath the stage, okay, there's the condenser, okay, and it's going to take that light from scattering, okay, around and kind of condense it and into a beam to shine through where you need to look, okay, and where you need to see. And so it's right up under here under the stage, okay, and then there's a dial. You can see that in the video. There's a dial here up underneath, like it looks like a ruler. Okay, and you're able to adjust that, okay, and that's going to adjust the amount of light coming through, okay, for you. And then at the bottom, we don't have it turned on, but this bottom is the light, right? There's a light, a bulb right in there, okay. And then you have this lever across the bottom, okay. This is the rheostat. So this whole thing is called the diaphragm, and it's called the iris, okay. Iris diaphragm lever. That's the last one right there. The iris diaphragm lever, okay? And what that's gonna do, if you think of iris, the color of your eye is the iris, okay? And as your pupil will open and close, so will the amount of light coming in and going, and not being able to go in, okay? And so as I would move this, and I'll get to demo it when we go to the microscope in a second, this will open up the beam to then be condensed, okay, to then shine through your specimen and into your eye, okay, a little bit more or a little bit less, okay, so be able to adjust it with those things, okay. Those are the basic parts, okay. Make sure that you go to your picture and that you label those parts, because you will have some questions where it just has a pointer, right, and if I go like this, and the question would be, identify the structure labeled A. And then you would have to write in, okay, or answer ocular lens, right? That's the ocular lens, okay? Or any other part that we go through. So just make sure that you practice that and that you have labeled those parts of that microscope, okay? All right, so I'm going to erase that, and we're going to go and talk about magnification. All right, so, and right after this, I'll take the microscope down, okay? Because I'm going to go over the magnification of these. So right after this, I'll take the microscope out of the way, okay? Can we see that? All right. Okay. Sorry. Videographer moments here. Okay. All right. So the next thing we're going to do is determine total magnification. So I said when we were talking about the parts of the microscope that the eyepiece, the ocular lens, magnifies it 10 times. Okay. So the ocular lens magnifies it 10 times. Okay. So I can put 10x, 10 times, on all of those. It's the same ocular lens, okay? If I go to the objective lens, okay, the smallest one, the little one, the scanning objective, okay, the scanning lens, okay, on it, it has a 4x. It says it right on it, okay? It magnifies 4 times. So if I was doing total magnification, when I have the smallest scanning objective in place, 10 times 4 is what? 10 times 4 is 40x, okay? I get all focused, I've adjusted everything I need to adjust, and now I put the next objective in place. This is the low power. It's the middle size one, it's the low power, and on the side of it, it has 10x. What is the total magnification, excuse me, of the low power with 10 times 10? That object is going to be magnified 10 times 10, which would be 100 times. Okay? Go to the next one, and on the side of it, it has 40. How much magnification, what is the total magnification of the object that you're looking at if you have the high-powered lens in place? The eyepiece is 10, the objective is 40. Total magnification is 400, hopefully is what you said, and you were waiting for me to say it for a while, right? But I was giving you time to do your math, okay? And so you would see that whatever I was looking at, when I looked at it through the microscope, the eyepiece and this high power would be 400 times bigger than what it would be if I just had it not in the microscope, okay? So I would be able to see it in much more detail, okay? All right, so I'm gonna take this down. Move the table out of the way. All right, so now let's look at, I said it's going to look 10 times bigger, and I have this little section added in. Talking about the field of view, okay? So the field of view is when I look in the microscope, that circle that I see is my field of view. That's what I view in that circle. So let's say that that circle up there is the field of view when you look in the microscope, okay? You could put a ruler across that and measure it, okay? 1 millimeter, 2 millimeters, whatever, okay? And you can measure that, okay? And so it's the diameter from one side of the circle that you see when you look in to the other side, okay? That's the field of view. Why is that important? Well, you want to know how big your specimen actually is, okay? And then it's also important as we go through those 3 objective lenses, okay? The field of view can help us estimate the size of our specimen, okay? So if I look in there and I know the field of view from one side to the other is 2 millimeters, and there's 1, 2, 3, 4, 5, 6, 7, 8 cells across, how big is each cell? Right? If we didn't know the field of view, we wouldn't be able to then estimate. If it takes 8 of them to fill it up, how big is each one? Okay? So it's in this circle, but I also wrote it out right here. Okay? So if the field of view is 2 millimeters and there are 8 cells visible, what's the size of each cell? 2 millimeters and 8 of them fit, 0.25 millimeters. So each one, 0.25, 0.25, 0.25, 0.25, okay? And going across, okay? And here's another conversion practice. Millimeters to micrometers. Here's another conversion practice, okay? So each cell is 0.25 millimeters, okay? How many micrometers is that, okay? We're going to move that 3 places, through that decimal 3 places, 1, 2, 3, to make it 250 micrometers, okay? So it's important to know what the field of view is so you can estimate the size of what you actually are looking at, okay? All right, so like I said when we were doing parts of a microscope, you're going to start with the scanning objective, the smallest one, okay? It has the largest field of view, okay? And you're going to be able to see the most to find what you're looking for, okay? So I'm going to switch to the microscope that's here on our table, and I have a slide already on there. It takes just a second to convert over. I have to hit the right button. Okay, so I have a slide of the letter E, okay, already on here, okay, for us to look at. This is the scanning objective. I'm going to write in black because the green probably is not going to show up on here. This is the scanning objective. I'm just going to abbreviate that, okay. The scanning objective is 4x, okay, but we also have to look through the eyepiece, okay. So it's going to go through the eyepiece, the ocular lens is 10x. So this E that you see is 40 times bigger than if I was looking at it on the slide. Okay? This isn't it, that's why I held it really fast. Okay? You can find it small. So it would be 40 times bigger than what would be if I'm trying to see it on here. Okay? So we can see that. Alright? And so on your paper, okay, you're going to need to draw what this looks like. Okay? So draw an E, okay, but you're also going to have quite a bit of space around it. Okay? Quite a bit of space around it. The other thing that's really cool about this compound light microscope is it actually reflects the image to you. Okay? And so it actually, when I look at that E on the slide versus up here, it's actually inverted. Okay? So it's like a mirror image for us. Okay? All right. So this is going to be 40 times bigger than what it normally is. Okay? So you're going to need to draw that. Okay? On there. So I'll give you a second to do that. And then the next thing on our lab is to center that E, and so I can put that in the middle. Oops, I went too far. Oh, no, I want to go that way. Okay, I would use those knobs on the side. I did that on purpose, right? You'll use those knobs on the side to move it where I want it to be. Okay, so I'm going to try to get it in the middle. Go up just a little bit. Okay, and then I'm going to use a piece that we didn't talk about, the nose piece. It's a black ring holding the objectives. And I'm going to put the low power in place. Okay? So now I have the low power, the middle size one in place. Put low anything. I know it's off the screen, but I didn't move anything, but we can see it's much bigger, okay? The ocular lens is 10x, and the low power is 10x. Total magnification, 100. 100 times bigger than if I looked at it with my unaided eye on the slide, okay? So draw that. So inside of your circle on your lab, draw the E almost touching top and bottom, all right? Or touching top and bottom, okay? Our screen looks square because it goes through a projector, okay? And that projector ends up being a rectangle, okay? And then you have a circle to draw. So just make that a circle and then draw that E. It takes up almost the whole space, okay? So while you're doing that, I'm going to erase this part. And then we would put the high power in, and that's really blurry, right? And so I would use the fine focus only to go, oh no, I went past. Okay, I have to go kind of slow to focus and try to find an easy edge. I might need to turn the light down. Okay, where you can see, we're only seeing part of that. Okay, if I was to gently move this, we can see that we're only seeing part of the top of that. So here and there. So it would be the top loop of the eye of the E, and then the hook at the bottom is off. Okay. So the ocular lens is still 10x, and now we're on high power, and that's 40x. Total magnification is 400 times. Okay. So I could try to see if I can find it, see it better, try to play with some stuff here, okay? But once we get there, we try to get a little bit of focus. We can see I'm going right past it, okay? But we're gonna have to try to see some detail. So if I want to say, is there something on the edge? Is there cilia? Is there microvilli? Like, is there something there? I would want to go on the highest power I could to see this edge, okay? And that would be the high power 400 times bigger, okay? Back. All right, so on your paper, you drew, okay, in your 3 circles, your field of view, okay, what that E looked like on the scanning objective, on the low power, and on the high power. So it should look something like that. Should look something like that, where there's space around it. Okay, maybe more space than that, but sort of some space around it for the scanning. Okay, remember that's where you'll start. It has the largest field of view and the lowest magnification. And then for the next one, the low power, 100 time magnification. Okay. Right here, see a little bit more detail, and then your last one should have looked something like that. Just a little bit. Okay, something like this, where the E, you're not seeing the entire E anymore because it's magnified so much. All right. All right. So most of these I answered while we were talking. So. As you increase the magnification, the field of view will do what? So this is a relationship. So as I increase the magnification, as I go from 40x to 400x, increasing the magnification, okay? What does my field of view do? It gets smaller, it decreases. So I can see the whole E and space, big field of view, right? I can see the whole thing, okay? But as I increase the magnification, I can only see part of it, okay? So right here, as I increase the magnification, as I increase the magnification, the field of view will decrease. Okay? It gets smaller, it gets more narrow, Okay, I'm fine tuning for right there. Okay, a little more pinpoint. Okay, which objective has the largest field of view? Which one of those E's did you see the whole E and space around it? The first one, right? So that is the scanning lens or objective. Okay? Which objective has the smallest field of view but the highest magnification? Highest magnification? Largest, smallest field of view. That would be the high power. Which objectives would you use to find the object first? So which one do you start with? Okay, I don't even know where it is on that slide, so I need to be able to scan the slide to find it. Okay, so which objective would I use? Which lens would I use? I need to scan to find it. So I need to use the scanning. Okay, scanning lens or scanning objective. Okay, so that's where I need to find the specimen first. All right, because we are not in class, we are not going to be able to do the wet mount. Okay, so we would have gotten pond water, and we would have gotten some pond scum, some algae in the bottom, and put a drop on that, and then we would try to hunt and find some protists or whatever we could find in there. Okay. So we'll be able to do that, but we do need to know this last question, okay? Do you always need to use the highest amount of light, the highest illumination, when observing a specimen? And the answer will be, let me go here, when we're looking at this. So I'm in the middle of the adjustment at the bottom was the rheostat, okay? Opening and closing that iris diaphragm, okay? And so if I close it, obviously I need some more illumination, right? But if I get to here, there's not too much. I'm about halfway, I'm about 3/4 of the way, I'm all the way. There's not that much difference between, okay? And so sometimes if you put too much light, if it's something that is not bold like this E, maybe like a protist, where you can kind of see through them a little bit, you can wash that out. It'd be too bright, okay? And so you want to be able to see your specimen in detail without washing it out, okay? Without being too dark on the other extreme, okay? And you also want to be what's comfortable for your eyes, because if you look at a microscope with the brightest light the whole time, it's going to get a headache. Right? That's when I would walk around and help students in class. It'd be the first thing I look at. I'd look and see how much light, because my eyes are very sensitive to light, and so I would turn it down, and then as I'm looking, I would adjust that a little bit. So it's not always important. It's not always required, okay, or necessary to have the highest amount of light. Okay? No, you don't always need the most light. You need enough light. And there's kind of variable in it, okay? So the microscope lab is pretty short. Parts of a microscope, total magnification, field of view, okay? And then which one has the most field of view or the least, okay? And then the illumination question, okay? Thank you, and make sure you email your professor if you have questions.